Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on IMViC test. IMViC stands for Indole, Methyl Red, VOGES-PROSKAUER, and Citrate Utilization Test. Indole test. Indole test is used to determine the ability of an organism to split amino acid tryptophan to form the compound indole. Tryptophan is hydrolyzed by tryptophanase to produce three possible end products, one of which is indole. Indole production is detected by COVEX reagent. This reacts with indole to produce a red-colored compound. Media and reagents used: peptone water broth and COVEX reagent. Procedure: inoculate the tube by taking the bacterial culture into peptone water. Incubate the tube at 37 degrees Celsius for 24 to 28 hours. Add 0.5 ml of COVEX reagent to the broth culture. Observe for the presence or absence of ring. Observation: in positive test, pink-colored ring after addition of COVEX reagent is seen. Example, Escherichia coli. In negative test, no color change even after the addition of COVEX reagent. Example, Klebsiella pneumoniae. Methyl red test. Some bacteria have the ability to utilize glucose and convert it to a stable acid like lactic acid, acetic acid, and formic acid as the end product. In the methyl red test, the test bacteria is grown in a broth medium containing glucose. If the bacteria have the ability to utilize glucose with production of a stable acid, the acid so produced decreases the pH and is indicated by a change in the color of methyl red from yellow to red. Media and reagents used: MRVP broth and methyl red solution. Procedure: inoculate the 2 tubes containing MRVP broth with a pure culture of the microorganism under investigation. Incubate at 35 degrees Celsius for up to 24 hours. Add about 5 drops of the methyl red indicator solution to the tube. A positive reaction is indicated if the color of the medium changes to red within a few minutes. Observation: MR positive. When the culture medium turns red after the addition of methyl red because of a pH or below 4.4 from the fermentation of glucose. Example, Escherichia coli. MR negative. When the culture medium remains yellow, which occurs when less acid is produced from the fermentation of glucose. Example, Klebsiella pneumoniae. VOGES-PROSKAUER test. The VOGES-PROSKAUER test is used to determine if an organism produces acetyl-methylcarbinol from glucose fermentation. If present, acetyl-methylcarbinol is converted to diacetyl in the presence of alpha-naphthol, strong alkali, and atmospheric oxygen. The diacetyl and guanidine-containing compound from the peptones of the broth then condenses to form a pinkish red polymer. Media and reagents used: MRVP broth, 5% alpha-naphthol, 40% potassium hydroxide. Procedure: inoculate a tube of MRVP broth with a pure culture of the test organism. Incubate for 24 hours at 35 degrees Celsius. Add 3 drops of 5% alpha-naphthol, followed by 1 drop of 40% potassium hydroxide. Shake the tube gently to expose the medium to atmospheric oxygen and allow the tube to remain undisturbed for 10 to 15 minutes. Observation: in positive test, a pink-red color at the surface is formed. Example, Klebsiella. In negative test, there is lack of a pink-red color. Example, Escherichia coli. Citrate utilization test. Citrate agar is used to test an organism's ability to utilize citrate as a source of energy. The medium contains citrate as a source of carbon and inorganic ammonium salt as the sole source of nitrogen. Bacteria that can grow on this medium produces an enzyme, citrate permease, capable of converting citrate to pyruvate. Pyruvate can then enter the organism's metabolic cycle for the production of energy. When the bacteria metabolize citrate, the ammonium salts are broken down to ammonia, which increases alkalinity. The shift in the pH turns the bromothymol blue indicator in the medium from green to blue. The media used in citrate utilization tests are Simon citrate agar procedure. Streak the slant back and forth with a light inoculum peak from the center of a well-isolated colony. Incubate aerobically at 35 degrees Celsius to 37 degrees Celsius for about 24 hours. Observe a color change from green to blue along the slant. Observation. In positive tests, growth with color change from green to intense blue along the slant is seen. Example, Salmonella, Klebsiella. In negative tests, no growth and no color change is seen. Slant remains green. Example, Escherichia and Shigella.